Thank you very much, Professor Basu, for organizing this symposium, and to Professor Majumdar for supporting this as president of the Indian Academy of Sciences. So what I'd like to do this morning or this evening, depending on where you are, is to tell you about some of the work that my research group is doing using material technologies to gain new mechanistic insights and take first steps towards translational progress in invasive brain tumors. Let me start with a bit of introduction about my research group. Most of what we do focuses on understanding or controlling mechanobiology at the cell matrix interface. We're very interested in how cells and materials interact with one another, and in particular, how mechanical and other physical cues from materials influence cell behavior. I'll show some examples of some of the work that my lab has done over the past 15, 16 years, and then really focus on our work in brain tumors. For example, we've been very interested in fundamental mechanisms that relate cell shape and structure to mechanics of cytoskeletal structures within cells. Here's an example where we used a very materials-oriented approach, single-cell micropatterning, to create extracellular matrix adhesive islands of defined size and shape, culture single cells on these islands, and use a single-cell technology called femtosecond laser ablation, which I'll talk about a bit later, to measure the mechanical properties of individual actomyosin stress fibers in these cells. We've recently partnered with Richard Harlan's lab at the University of California, Berkeley, to understand how the interplay between the mechanics of basement membrane and skin and mechanical forces that skin cells generate, how the interplay can give rise to patterning that forms within skin, in particular, the pattern of hair and feathers. We also have a very exciting new collaboration with Professor Andreas Stahl here at UC Berkeley, examining the role of mechanical forces in governing metabolism of brown fat cells with an eye towards developing strategies to enhance thermogenesis of these tissues. And finally, we do a bit of forward material design where we develop polymeric systems that derive their design principles from biological systems. One thing we've done recently is to take cytoskeletal networks that stabilize the structure of axons and develop new materials and stimulus-responsive surfaces that are based on the proteins that are found within cells. And so here's an example where we can reversibly swell and condense one of these protein systems by using enzymatic phosphorylation as a switch. What I'd like to focus on in today's presentation is our effort to use material technologies to understand, control, and model the spread of glioblastoma, which is a very aggressive brain tumor. Glioblastoma, or GBM, also known as grade 4 astrocytoma, is a very, very aggressive and almost uniformly fatal brain tumor. It's the most common primary intracranial tumor. It's a relatively rare tumor, but it is very compelling to study because it has a very aggressive clinical course. So the median survival with this disease is only about 12 to 15 months. And this is with aggressive surgical resection, radiation, and various types of targeted chemotherapies. This series of magnetic resonance images on the left side of the screen illustrates the aggressive course of GBM. So first presentation of GBM might be accompanied by psychological changes or persistent headaches or unexplained seizures. An MRI might reveal this mass lesion, as you see here, in the right cerebral hemisphere. First line treatment is surgical resection to reduce intracranial pressure, as well as radiation and temozolomide, which is an anti-mitotic agent, to try to reduce tumor progression. What will happen almost inevitably is that several months later, patients will return with recurrence of tumor. And you can see these hyperintensities surrounding the resection cavity and starting to make their way into distal regions of the brain. And then by 17 months, and this happens to be a post-mortem scan, you can see extensive infiltration of brain tissue by the tumor. And one of the truly devastating things about this disease is that despite all of our advances in surgical care, radiation, and chemotherapy, this median survival time has not really improved very much, even over the past century. So there's a great deal of interest in trying to understand how tumor cells infiltrate brain tissue, both as a way of gaining greater insight into the pathophysiology of disease, but also as a way of helping to make drugs that do make a difference work better. Here's an example of the latter concept on the right. Anti-angiogenic agents such as bevacizumab or Avastin have received a lot of attention in brain cancer and other cancers because of their ability to choke off the blood supply of growing tumor. And indeed, in GBM, anti-angiogenic agents do a good job of shrinking tumors, at least initially. Unfortunately, over time, the tumor cells will acquire what's known as evasive resistance and start to migrate very, very aggressively into brain parenchyma to find blood vessels, which in a lot of ways makes sense conceptually. If you choke off the tumor's blood supply, it will respond by seeking out its own blood supply, which requires migration through the brain. So there's this idea in the field that if one could couple anti-angiogenic agents with agents that block the invasion process, one might achieve a synergistic effect. We first became interested in this problem about 15 or 16 years ago, in part because we became aware that there are some mechanical changes associated with tumor progression in GBM. One of them is that, as with many tumors, clinicians will tell you the tumors are significantly stiffer than the surrounding tissue. And this has been formally shown in this ultrasound study shown here, where, without getting into too much detail, ultrasound turns out to be more sensitive than either T1 or T2 weighted MRI of predicting the margins of a tumor, which indicates that tumor growth is associated with changes in stiffness and density in the tissue. In addition, GBM tumors don't spread isotropically or randomly within brain tissue. They tend to follow very specific anatomic paths, as I'll discuss later. And one of the fastest routes of spread of these tumors is to follow vasculature. And vasculature is significantly stiffer, about an order of magnitude stiffer than the surrounding brain parenchyma, which is comparatively soft. And so there's this idea that perhaps tumor cells might be using blood vessels as contact guidance cues to mediate their very rapid invasion throughout brain tissue. Our first foray into this problem back in the late 2000s was focused on asking some very simple reductionist questions. And the first question we asked was, if you challenge brain tumor cells with polymeric matrices that have defined differences in stiffness, does this influence behaviors in culture that might be relevant to their growth and spread in vivo? So these are polyacrylamide matrices that have been conjugated with a protein called fibronectin. These are human brain tumor cells, a cell line called U251, cultured on these materials. And you can see that on relatively stiff matrices, the cells spread very nicely, engage the matrix, are capable of moving in a very smooth fashion. And as you go to softer and softer materials, eventually the migration stops, and it turns out proliferation reduces as well. The upshot or summary of this work is that soft matrix appears to be a stop signal for migration. And so a corollary to this is that perhaps tumor cells have to remodel the extracellular matrix to make it stiffer, as is observed clinically, in order to migrate. These sorts of hypotheses about material properties and how they regulate in vivo progression can be very difficult to directly test, in part because it's hard to control the material properties of tissue. However, we can manipulate some of the signaling systems that recognize changes in stiffness and ask whether manipulation of those systems influences tumor progression. And so we did that very experiment a few years later, where we overexpressed a constitutively active version of a protein called Rho-A, which is very important for sensing mechanical signals. And I'm not showing the data here, but we showed in this publication in Cancer Research that when you do this, you're able to sensitize human brain tumor cells to soft matrix, more sensitive than they otherwise would be. And then if you take those transduced cells and inject them into an immunocompromised mouse, you still form tumors, but the tumors are much less infiltrative, and the mice survive about 30% longer than they otherwise would. So it suggests that manipulating the systems that regulate mechanosensing can indeed make a difference in terms of limiting proliferation and extending survival. I'll mention that since this work came out, a number of other groups have very creatively directly demonstrated some of the aspects of our hypothesis. So this group at the University of Munster in Germany used a Drosophila brain tumor model to show that as tumors progress, they engage integrins and they begin to secrete enzymes, in particular lysyl oxidases, that covalently cross-link extracellular matrix proteins, thereby stiffening the extracellular matrix. And this in turn gives rise to a feedback loop that promotes tumor progression. This more recent study from Ji Huang's lab at the University of Toronto, published in Neuron a couple of years ago, implicated an ion channel called piezo-1 in this process, and essentially showed that as tumors progress, they stiffen the extracellular matrix. This leads to heightened signaling through this ion channel piezo, and that in turn turns on programs that promote rapid invasion. So I encourage you to check out those two papers if you're interested in exploration of this hypothesis in more detail. What I'd like to spend the rest of my time talking about are our efforts to build more anatomy into these models. As I mentioned, glioblastoma tumors don't spread radially isotropically within brain tissue. They tend to follow preferred anatomic tracks. And to a first approximation, there are three routes through which GBM spread clinically. And I'll mention that this formulation was really strongly inspired by a wonderful collaboration we have with a neurosurgeon at UCSF, Manish Aghi, who does a lot of GBM cases and thinks critically about how GBM tumors might respond to anti-angiogenic agents. So one mode of spread is called peritumoral, where tumor cells are essentially tunneling their way through the brain parenchyma. The main extracellular matrix components in play here are polysaccharides, and in particular, a polysaccharide I'll talk about a lot later called hyaluronic acid. A second mode of invasion is along white matter tract, so this is following axons within the brain. Here's an example shown down here. And then the third mode, which I mentioned earlier, is perivascular invasion, where tumor cells will home towards blood vessels and then very rapidly migrate along those vessels. And it's these two modes of migration that are very, very fast. So we've been interested in trying to apply materials technologies to model each of these modes of migration. As I mentioned, hyaluronic acid, or HA, is the main component at play in parenchymal or peritumoral invasion. Here's a schematic diagram illustrating a cell in contact with a hyaluronic acid matrix. The blue here would be HA. HA in tissue is cross-linked by a variety of proteins, most notably tenascin R and tenascin C. In addition, hyaluronic acid can directly bind receptors on the cell surface, most notably CD44, which is closely associated with tumor progression. Hyaluronic acid comes up quite a lot in musculoskeletal biology as well. It's the main matrix component of cartilage, and so much of what we know about the biology of HA crosses over between these two fields. One of the things that's nice about hyaluronic acid is that you can manufacture it or create hydrogels from it in the laboratory. So hyaluronic acid, you can see here, is this linear polysaccharide chain. It has a number of functional groups that can be used to do chemistry, including an acid group, an amide group, and a hydroxyl group. And so the way we form three-dimensional networks out of these materials in the laboratory is by using these chemical groups as functional handles to do cross-linking and peptide conjugation. So for example, we can functionalize the hyaluronic acid backbone with methacrylate groups, which then primes HA for Michael addition chemistry. So we can do, for example, we can add peptides that have cysteines in them and use the sulfhydryl groups in the cysteines to covalently link these peptides to the backbone. And sometimes these peptides have sequences that will bind cellular integrins to facilitate adhesion. We can also physically, rather chemically, cross-link these materials by using dithiol molecules, including dithiophenyletol. We've done PEG dithiols as well, and form a three-dimensional network of some desired stiffness. So we like these materials in part because the sort of migration that we see of tumor cells within these materials is very close to what others have observed in tissue. So here's a tumor cell tunneling its way through a three-dimensional HA network. This is the cell nucleus squeezing through pores in the matrix. These are dendritic processes the cell extends as it tries to move forward. And it looks very, very much like observations others have made of tumor cells migrating through tissue, in this case, a brain slice model from Steve Rosenfeld and Peter Knoll, published a few years ago. So I'm going to skip over a lot of the sort of proof of principle work that we've done with HA matrices and talk a little bit about our efforts to understand the biology of HA adhesion. But first, I'll just very briefly mention some of our efforts to use this as a translational platform. So one thing we became very interested in doing very early on was figuring out ways to miniaturize these hyaluronic acid matrices to make them amenable to imaging and to screening. And so we've deployed, and you can read about this in this publication in 2018, we've developed these micro devices where we can implant essentially tumors in a dish. They can tunnel through a three-dimensional hyaluronic acid matrix and make their way towards a channel that looks, at least topologically, like a blood vessel. And so we can follow the process of this peritumoral invasion and eventually perivascular invasion. And this very simple approach turns out to be quite powerful in terms of both understanding what factors of matrix control invasion, for example, the presence or absence of myosin contractility, the presence or absence of integrin binding peptides, as well as the responses of these tumors to drugs. And so in collaboration with Dr. Aghi's lab, our collaborator at UCSF, we've looked at the invasion of tumors that are very sensitive to bevacizumab and very resistant to bevacizumab. And you can see that the resistant tumors aggressively invade the hyaluronic acid matrices, much more so than the sensitive tumors do. And this is exactly what is observed clinically. So what I'd like to spend a few minutes talking about are our efforts to understand fundamental aspects of how tumor cells engage hyaluronic acid matrices. And what I'll tell you about is the work of Kayla Wolf, a former graduate student in the lab who's now doing a postdoctoral fellowship with Jennifer Lewis at Harvard University. So when Kayla did imaging of brain tumor cells on bare hyaluronic acid matrices, she found that they adhered to these matrices through a very distinct mechanism. This is the cell body right here, and if you look carefully, you can see these very long projections that extend many microns from the cell body, sticking out in all directions. This is something that we see for a number of continuous culture models. We also see it for patient-derived tumor cells that are a little bit closer to the original tumor. In all cases, they form these very long structures. These structures are very dynamic. If you use live cell imaging to follow their growth and retraction, what you find is that they are capable of extending and retracting over a period of minutes to tens of minutes, which is relatively rapid as these things go. To reassure ourselves, this was not simply an artifact of two-dimensional culture. We looked for these structures in three-dimensional HA matrices and were able to find them. We also looked for these structures in an organotypic brain slice where tumor cells were invading through live brain tissue. And we see these structures here as well. So we think that these structures are physiologically relevant. A question would be, what are these structures and how might they facilitate migration? So one obvious comparison is to structures called philopodia, which are these very long protrusions that are found in all kinds of migrating cells. We don't believe these are philopodia because these typically contain a myosin isoform called myosin 10. We don't see these in our structures. They resist disassembly by drugs that normally will disassemble philopodia. And most importantly, if you're a cytoskeletal biologist, these structures are very actin dependent. So if we treat philopodia with drugs that disassemble actin, it turns out they don't disassemble these processes. Perhaps a more apt analogy would be to structures called pseudopodia, which have been identified for several decades now. And you can think about these structures as almost being battering rams that enable cells to force their way through three-dimensional matrices, tend to be microtubule based and have a ring of actin. But probably the closest analogy are to structures called microtentacles, which were discovered by Stuart Martin at the University of Maryland and are a feature of circulating tumor cells. So they're often found in tumor cells that are in the vasculature prior to their metastasis. And it's thought that these structures act almost like Velcro in sticking to the endothelium as a first step towards invading that distant tissue. And so because of the structural similarities between these classically described microtentacles and what we saw, we just adopted the name and called these structures microtentacles as well. It turns out these microtentacles are coated with a protein called CD44 I mentioned earlier, which is an adhesion receptor for HA. So in a sense, it makes sense that these are facilitating adhesion to the hyaluronic acid surface. But to prove that formally, if you use small hairpin RNAs to knock down CD44, what we find is that we have almost complete loss of these microtentacle structures. And though I'm not showing it here, we have almost total abrogation of adhesion to the hyaluronic acid surface. To get some insight into what these microtentacles are doing, one question we wanted to ask is what sorts of forces do they bear? Are they, for example, bearing tensile forces that are generated by the cell body? And so a simple experiment that we did, which I'll talk in a little bit more detail later about, is called laser ablation. So we can take a femtosecond laser and physically sever one of these microtentacles. And in this experiment, we asked, what happens to the rest of the cell when we do this? What is the nature of the recoil? And you can see in this time lapse that when we cut this microtentacle located right here, the rest of the cell body retracts from left to right, which indicates that the structure was bearing tension, and then the release of that tension causes a reciprocal recoil of the cell body. Super resolution imaging has been very helpful in terms of understanding what's inside these structures. This is an example of what a cell sitting on a glass surface coated with integrin ligands might look like. You can see the microtubule network here in red, the actin network here in green. There's this well-described process of retrograde flow of the actin network. It polymerizes to push the front of the cell forward, and then myosin motors pull it back. We see something very different on hyaluronic acid matrices. You may or may not be able to appreciate this, but here's a single microtentacle, and we see both actin and microtubules within these structures. And additional imaging, this probably isn't the clearest example, indicates that microtubules seem to promote extension of these tentacles, and actin seems to pull it back. So there's almost this mechanical balance between these two cytoskeletal systems. So that raises the question, mechanically, how might the adhesive machinery, as exemplified by CD44, be coupled to actin and microtubules within these structures? And without getting into too much detail, we converged on this system involving association between an actin binding protein called, IQ-GAP1 and a microtubule binding protein called CLIP170. and did staining and showed that both of these proteins are present within these microtentacle structures, along with both actin and microtubules. And to skipping over a lot of data, we converged on this model in which microtubules have CLIP170 at the end of them, actin has IQ-GAP1, and these two proteins form a complex that enable coupling of microtubules and actin within these microtentacle structures. However, these microtentacles are only stabilized and are useful for adhesion to the matrix if this entire complex can engage CD44 and thereby mechanically couple these tentacles to the surrounding hyaluronic acid. And so if all of these components are present, you get productive formation of microtentacles and migration. To demonstrate whether these are functionally important, we use CRISPR-based gene editing to knock out IQ-GAP1. And very satisfyingly, when we did this, we didn't see any effect on adhesion on RGD or integrin ligating matrices. But when we looked at HA matrices, which can only bind cells through CD44 and other HA receptors, knocking out IQ-GAP1 leads to almost complete loss of adhesion. And similarly, we see loss of microtentacles. We also see significant reductions in migration speed on these matrices. Are these clinically relevant? Well, we can take a panel of human patient-derived glioma stem cell lines and ask about their expression about IQ-GAP1 and CD44. And what we find is that the most aggressive tumors clinically, those that fall under a subtype known as mesenchymal, tend to have the highest expression of both of these proteins. If we examine larger databases, in particular the Cancer Genome Atlas, which has population-level data from many, many tumors, we also see this broad association between tumor aggressiveness and expression of these two proteins. And finally, if we look at survival of patients that have various levels of expression of each of these proteins, we find a strong correlation between high expression of CD44 and CLIP170 and survival of the patient. So it seems that this has some clinical correlation as well, and we're currently working up these targets to see if they might be useful clinical targets. This work was published just a few months ago in PNAS and sort of describes both the basic biology of these structures as well as some of this more clinically facing data that I just mentioned. So what I'd like to do is to shift gears for the rest of the time that I have and talk about the other modes of invasion. So we focused on peritumoral invasion, where cells are tunneling their way through HA. Now what I'd like to do is to turn my attention to white matter tract and perivascular invasion. What these two share in common is that in both cases, the tumor cells are following some sort of structural cue, whether that's the axons found within white matter or the blood vessels in vasculature. So here's some schematic diagrams, of what these two types of migration look like. And what both of these modes of migration have in common is that they can be conceptualized as migration of a tumor cell within a confined one-dimensional space. Here's a cartoon showing tumor cells migrating along white matter tracts, and you can see in this close-up that the tumor cell is essentially sandwiched or confined between the axon of a nerve cell and various extracellular matrix components. So it really has this restricted space in which to move, which gives rise to fast invasion along that axis. And you can really see this in tissue sections where there's almost this pocket where tumor cells migrate along axons. The same is true of blood vessels. Beautiful intravital imaging, in this case from Frank Winkler, 2009, published in Glia, shows tumor cells migrating along blood vessels. And in this paper, they had a very, very nice correlation between the number of vessels that are contacted and the speed of migration. In particular, glioma cells that are capable of interacting with multiple blood vessels migrate much more rapidly, which sort of further reinforces this idea that these structures are being used as contact guidance cues. So this raises the question of how to model this geometry in the laboratory. We had a history, even before focusing on this problem specifically, of using microfabricated microchannels as a model for studying confined migration. This is the work of Amit Pathak, who is a former postdoc within the lab and now is on the faculty at Washington University in St. Louis. And he was interested several years ago in trying to dissect the effects of matrix stiffness or mechanics and matrix confinement on governing cell migration. So what he did was made this polyacrylamide-based microfabricated scaffold where he used the typical tools of soft lithography to make silicon masters with defined topographical features, and then molded polyacrylamide matrices around these silicon masters to give rise to these soft materials or soft surfaces that had three-dimensional channels in them and that were amenable to cell culture. And so he both dissected the effects of matrix stiffness and confinement on cell migration, as well as used platforms like this to understand how defined genetic lesions within this breast cancer progression series influence sensitivity of migration speed to each of these parameters. And these studies were published in these papers shown here. We've also combined these microchannel platforms with single cell analysis. So this slide is the result of a collaboration with Professor Amy Herr in my department at Berkeley, who's one of the world's experts on single cell proteomics, in particular single cell Western blotting. And so in this paper published in Lab on a Chip a few years ago, we combined microchannel-based migration assays with single cell Western blotting. So we could take a complex mixture of cells, force them to migrate within one of these channels, the idea being that the faster cells will migrate further along the channel, and then do single cell Western blotting within the walls of the device to measure expression of specific proteins. The basic idea is to correlate the migration speed or some measure of motility against expression of some protein. And to an extent, we were able to put together a panel or signature of expression of specific proteins that were predictive of very aggressive migration and seemed to line up quite well with what is observed clinically. So a few years ago, Carmela Rihanna, a postdoc from the University of Bremen in Germany, came to our lab on a DAAD fellowship for a year and became interested in using these microchannel platforms to understand something about how the mechanics of cells change as they undergo this confined migration. I'll skip over a lot of detail here. She essentially made these PDMS-based devices. So this is now an open channel device where the medium is on top of the device. It's fully accessible to surface technologies. And you can see that as cells encounter the mouth of this device, they sort of go from fully unconfined migration to associating with one wall. And then when they finally enter this device shown here, and you can see this in schematic form on the left, they undergo this fully confined migration. And as others have observed, and consistent with clinical data, when cells are in this confined migration, because they only have one direction to go, they move in a very rapid and persistent fashion. And that's something that we can see in these devices as well. So Carmela was very interested, as I mentioned, in understanding how mechanics of these cells might change. And in particular, she wanted to take on a longstanding open question in the field, which has to do with whether tumor cells have to soften in order to migrate through confined spaces, perhaps to be able to rapidly deform themselves. And so this is where the open channel device design came in. We wanted to have a design where we could directly interrogate the cells in these various states of confinement using atomic force microscopy, which requires direct contact between this force probe and the surface of the cell. And so this is the experiment that she did. She used AFM both to image cells in these various states of confinement, as well as to measure mechanics of these cells. So in particular, did indentational mechanics measurements on cells and use the resulting indentation curve to extract a Young's modulus for the cells, a measure of stiffness. So here's a plot of Young's modulus or elasticity as a function of degree of confinement. A is unconfined, B is associated with one wall, C is beginning to enter the channel, and D is fully confined. And the broad pattern here is that even though there's quite a lot of spread in the data reflecting the heterogeneity of the cells, as cells become increasingly confined, they become softer and softer, consistent with this hypothesis that cells must soften in order to deform themselves through very tight spaces, but to our knowledge, the first direct demonstration of this idea. These changes in mechanics are also associated with changes in signaling, typically associated with mechanotransduction. In particular, this protein, yes-associated protein, or YAP, which is part of the Hippo pathway and has been implicated as a master regulator of mechanotransduction, changes its localization in cells in very characteristic ways as a function of confinement. So this is the unconfined case. Cells have not yet entered the channels, and you should be able to see that YAP primarily localizes to the nucleus. There's overlap between the YAP signal and the DAPI signal, which is a marker of nuclear DNA. And this is consistent with what one would expect to see on a very stiff surface. However, as the cells enter the channel and soften, which we demonstrated by AFM, this is accompanied by expulsion of YAP from the nucleus. So we no longer see nuclear localization of YAP, consistent with the idea that the cells are softening, and also suggesting that the softening is functionally important because YAP is a transcriptional coactivator and has been known to activate a set of genes responsible for the stiff gel phenotype. So this work was published in Molecular Biology of the Cell just a few weeks ago, if you're interested in reading more about it. One of the findings that we had that I didn't spend a lot of time talking about was how ultrastructure changes as these cells move through various states of confinement. This is super resolution imaging of the actin cytoskeleton in these various states of confinement. And when the cell is completely unconfined, and I'll show better examples of this in a moment, you can see formation of these networks of what are called stress fibers, which are bundles of actin, myosin, and actin binding proteins. Essentially the cell's tensile network that enables it to generate traction forces against the underlying material. Once the cell enters the channel, something very interesting happens. The network changes from this nice two-dimensional network of stress fibers to almost an aligned network of cables that all run in parallel. And as you make the channels narrower and narrower, eventually these stress fibers get redeployed to the walls of the channel as opposed to the floor. And this is very interesting because stress fibers are known to be important for mechanically polarizing the cell during migration. So as I mentioned, these structures are made out of this polymer actin. They also contain a motor protein called myosin and a number of actin-binding proteins. And so actin at the front of the cell can form these diffuse branch networks to enable protrusion of the cell forward, these contractile structures that give rise to retrograde flow and enable the cell to pull itself forward in the front of the cell. And they're very important in the rear of the cell as well. They tend to assemble into these very large, highly contractile bundles that facilitate retraction of the trailing edge of the cell. So you have these relatively weak, well-distributed forces at the front of the cell and these very strong and concentrated forces at the rear of the cell. And if you don't have both of these things happening, one does not have front-back polarization of the cell. So we've been really interested in asking, where does this come from and how do stress fiber networks develop mechanical polarization? And in addressing this question, a technique that's been very useful for us has been laser nanosurgery. I mentioned it earlier in the talk. So in that case, I was talking about severing a microtentacle. It turns out we can do this surgery, if you like, even more precisely. In fact, we can cut single stress fibers within living cells. Here's a movie that goes back all the way to my postdoc with Don Ingber back at Harvard Medical School showing severing of one of these stress fibers. And so the structure is under tension due to the myosin motors. And so when we sever it, that tension is unloaded and the ends retract back from one another, much like an elastic band. In addition to generating movies like this one, we can also quantify the data that we get, and in this case represent it as retraction distance as a function of time. And very satisfyingly, as an engineer, the data they fit very nicely to mechanical models that describe viscoelastic cables. This is a Kelvin-Voigt model, which is the simplest way you can describe such a structure, a viscous and elastic element in series, or in parallel, excuse me. And there are a couple of parameters that define this model. One is the total extent to which the structure retracts, which is a measure of stored elasticity. And then there's the time scale of retraction, which reflects the relative viscoelastic character. So for those mechanical engineers or material scientists in the audience, this is very much like a creep response of a viscoelastic material. And so we can do something with these parameters, use them as measures of the elastic state of these fibers. Stacy Lee, a former graduate student in the lab, used laser nanosurgery to systematically map the mechanics of each of these populations of fibers, published in this paper in 2018. And to make a long story short, she showed that there are characteristic differences in the elasticity of each of these populations of fibers. And in particular, stress fibers at the front of the cell bear less elastic energy than stress fibers at the rear of the cell, which sort of makes sense with this idea that I mentioned earlier, that stress fibers at the rear of the cell have to generate these very large forces to retract the trailing edge. So where does this come from? What molecular mechanisms might underlie development of this mechanical polarity? One candidate that we've been focusing on, and this is the last few slides I have before I finish, has to do with this cycle or pathway, including the proteins limb kinase and cofilin. So this is a picture of the limb kinase cofilin cycle. Cofilin is an actin severing protein, so it's capable of taking filamentous actin and chopping it up into small pieces. It can be phosphoinactivated by a number of kinases, most notably limb kinase. So it phosphorylates cofilin, can turn it off, and allows actin polymerization to proceed unimpeded. Cofilin can also be dephosphorylated by a number of phosphatases, including the slingshot proteins, chronophin, and others. And so that turns it into an active state and enables it to cut actin once more. So given that cofilin is very important in remodeling actin filaments, it occurred to us that maybe this pathway might be important in enforcing mechanical polarity. And so an analogy to think about that might help guide how we think about this has to do with pruning of various actomyosin cables in order to ensure strong ones at the rear of the cell, sort of like trimming of this bonsai tree shown here. The basic idea is that as you remove some of the low tension branches, in this case, the smaller branches, overall, the effect is to increase the strength of other branches. And so without getting into much of the background, we hypothesized that one thing that cofilin was doing at the front of the cell was essentially deselecting low tension fibers, such that by the time these stress fibers make their way to the rear of the cell, they bear very high tension. And so Stacy Lee, who I mentioned earlier, a former graduate student, did a number of experiments where she used shRNAs to knock down cofilin. The first thing that she found is that when she did this, the cells failed to polarize, kind of consistent with this idea. So rather than having a very clear front and back, as shown in these non-targeting cases, the cells are rounded, not rounded in the sense that they can't adhere to the matrix, but rounded in the sense that they're trying to extend the lamellipodia everywhere. Essentially, all of the cell is now the front. And this persists even if we treat cells with epidermal growth factor, which is normally a very strong stimulus of polarization and motility, and they are still unable to polarize in the presence of EGF. We can even culture cells on single cell micropatterns that look exactly like a polarized cell should look, with a well-defined front and back. Whereas the non-targeting cells behave and form a front and back where they should on these patterns, the knockdown, cofilin knockdown cells do not, and they attempt to extend lamellipodia everywhere. So this is live cell imaging showing what's happening to the stress fiber network in each of these cases. Here's a non-targeting cell showing well-defined transverse arcs and ventral stress fibers. In the case of cofilin knockdown, you can see that there are essentially arcs throughout the circumference of the cell, which again is consistent with the idea that the cell is trying to generate a front everywhere. Now, what's very interesting is the sorts of ultrastructures that these cells form. So as I mentioned, one of the characteristic stress fiber types at the front of the cell that stabilizes the front of the cell are these transverse arc structures. And they're very, very important in ensuring that the front of the cell has this lamellar shape. And you see normal formation of these transverse arcs in non-targeting cells and failure of that arc maturation when you knock down cofilin. And this is accompanied by changes in the types of proteins that are incorporated into these actin-based structures. So here's the non-targeting case first. As I mentioned, you have these very nice parallel arcs that eventually fuse into a mature arc. However, when you knock down cofilin, you instead get these geodesic structures that form, and I'll show you another example of this in the next slide, where instead of forming these parallel stacks of arcs, you instead get these two-dimensional networks of actin that are cross-linked by this protein alpha-actinin. Here's an even better example here, confocal and super resolution imaging showing these structures. I'll just maybe focus on this example here and this example here. So you can see, as I mentioned earlier, you get these geodesic structures that have nodes that are very rich in alpha-actinin. And in addition, these nodes exclude proteins that bind and are important for the function of myosin, the motor. So tropomyosin, which is a very prominent feature of these transverse arcs, is typically excluded from these geodesic nodes, almost indicating that the stress fiber network is trapped in this two-dimensional network state that precludes it from being able to generate high contractile forces necessary to establish front-back polarity. And so this is a model that we put together that is consistent with what I mentioned earlier, that what cofilin is doing is essentially pruning out the low-tension subunits that are found at the front of the cell and allowing mature arcs to form. And in the absence of cofilin, you have persistence of these non-contractile actin subunits. They make their way into what should be the maturing arc network. And instead, you get these two-dimensional geodesic structures that form, which, while very pretty to look at under the microscope, do a poor job at generating the contractile forces necessary to establish front-back polarity. And so this was published in the Journal of Cell Science earlier this year. So finally, to bring this full circle with GBM, as I mentioned, one of the key regulators of cofilin is this protein lim kinase, which turns cofilin off. And so you might wonder whether manipulation of this cycle might influence the progression of GBM in tissue, if indeed it's important, the system is important for confined migration. And so we did that experiment. We knocked down the two isoforms of lim kinase 1 and lim kinase 2. In human patient-derived glioma cells, characterized them in vitro, as well as implanted them into immunocompromised mice to look at progression in vivo. And indeed, we find that suppression of lim kinase reduces tumor growth, both in vivo as well as in vivo. And so you have smaller tumors, they're less infiltrative, and this translates, much like the row experiment I showed you earlier, into an extension of survival. And you can really see the difference in terms of the diffusiveness of the invasion when you look at tissue sections. So just to sort of quickly summarize what I've talked about, this is my last slide. We've tried to bring the mindset and the toolbox of material science to dissect these various modes of migration in GBM, including three-dimensional high hyaluronic acid networks to look at peritumoral invasion, and these confined migration systems to examine and dissect perivascular and white matter tract invasion. And so now we're at the stage of not only asking whether lessons we learn from these reductionist studies matter in vivo, but also whether there might be actionable targets that could be used therapeutically. And so I'll refer you to this review that a number of members of my lab collaborated on that was published in Nature Reviews Materials last year that talk about the material science of the glioblastoma microenvironment. So I'd like to stop and thank the folks that did the work. Kayla Wolf is a former graduate student, now a postdoc at Harvard, who led all the microtentacle studies. Carmela Rihanna did the work of measuring stiffness of cells in confined spaces. She's now doing a postdoctoral fellowship with Tillman Schaefer at the University of Tübingen in Germany. And Stacy Lee, a former graduate student in the lab, who did all of the cofilin work and is now working in industry at Emerald Cloud Lab. Here's a photo of our lab, I guess in the Zoom era, which is as close as we can get to a group photo these days. So the work, as I mentioned, a lot of this is done in collaboration. Much of our tumor work we do now in collaboration with Manish Aghi's lab. Ulrich Schwartz has been an important partner for much of the stress fiber mechanobiology work and some of the other names that I've tried to credit along the way. Here are funding sources that support the work. So I'll stop here. I'm more than happy to take whatever questions you might have, and my sincere thanks again to the organizers and to the Academy for the opportunity to speak.